Infections are a big problem worldwide. They're probably still the biggest killers of human beings, and not only do they kill people, they also cause a lot of what we call as morbidity, which means that people get disabled and other things happen. So there are lots of infections. One of the infections that we work on predominantly are bacteria, and one bacteria that we really work on specifically is M. tuberculosis, or the bug that causes TB. Now, just to give you an idea, this is just one bug, and this one bug is causing about 10 million new cases of TB every year. Now, if you combine this with other bacteria, the other bacteria, just name a few Staph aureus, or people call it MRSA. These are killing a lot of people, not just globally, but in the United States itself. And it's a big problem. It costs us billions of dollars in healthcare, and we don't have very rapid ways of diagnosing.
Infectious diseases is a really important issue that is growing. It's getting more complicated. Bacteria are getting resistant, and we need new innovative tools to be able to determine treatment and visualize outcome in the patients as soon as possible.
Imaging infectious diseases is very interesting using PET-MR, so that we can image the living organism in another living organism. So having the host and the pathogen, and then consecutive imaging allows the dynamic imaging of the progression of the disease.
We're imaging infectious diseases, and the purpose is to try to find tracers that are specific for bacteria or for fungi in animal models that are representative of the infections that we see in humans. We develop those tracers in such a way that they can go from different modalities. If we can do optical imaging, if we can do SPECT, if we can do PET, whatever fills the situation that we need from the animal model and from the tracer. We have a significant amount of people out there that need proper diagnosis and proper monitoring of infections. If we can do it that non-invasively, it's a huge advantage. Currently, what we do is to poke a needle into the tissue that is infected and try to take out the bacteria, culture it, try to grow it outside to be able to characterize what it is. That is invasive. That leads to a lot of risks. If we could do that using imaging, that is the future.
The WMIS has an interest group which is focused on imaging. It's called the Imaging of Infection Interest Group, and I chair that. The interest group is very important because we want people, brilliant people like you who are involved in imaging, to focus on infectious diseases, which I think is a big issue worldwide. Bacteria as well as viruses and other pathogens or bugs can be in our body in various states. They could actually be multiplying actively and then causing what we would call as active disease, where people would have fever and they would have symptoms. But they can also live and hide inside our bodies in a kind of a quote-unquote dormant or sleeping state. And it's advantageous for the bug to do that, right? Because it's not causing any symptoms. Everybody is going about, we're not trying to kill them. And they will then come back many months, sometimes decades later. It's sometimes difficult to be clearly able to differentiate by testing. I mean, of course, if the bugs are dormant, you're not going to have these symptoms, and that's a pretty easy sell. But sometimes it's difficult to know the people who might have the dormant bugs, which might just turn up at some other time of their lifetime, especially at the times when they don't want it to be coming up, when they're immunosuppressed, when they're getting cancer chemotherapy and things like that.
So we try to image the pathogen itself via antibodies or other targets, and then compare this with the unspecific tracers, for example, the glucose marker FDG.
So that's the reason I love imaging, and it's non-invasive. It will give you information from any part of the body, and it's relatively rapid.
So we developed an analog of sorbitol, fluorodeoxysorbitol, that is derived from glucose in a simple 30-minute reaction, and it's used to differentiate between gram-positives and gram-negative bacteria. Once you have a patient that has an infection, you want to classify whether the bacteria is a gram-positive or gram-negative to try to streamline the antibiotic choice that you're going to use in the patient. This probe called FDS allows us to image. It accumulates within the enterobacteria specifically, and we're able to visualize that bacteria using PET scan. We did models for E. coli thigh infection, we did E. coli in the brain tissue, Klebsiella pneumonia in the lung, and it's beautiful how it accumulates, and we can track the infection and monitor the antibiotic response in those animal models. We showed in mice that once we infected them with drug-resistant E. coli and drug-susceptible E. coli, imaged them at time 0, then gave antibiotic treatment for 24 hours. We imaged them again at the end of that period. We saw that the susceptible bacteria to the antibiotic disappeared, the signal disappeared. The ones that were resistant, the signal was still there. So what if you could do that with humans, finding out that the bacteria is still there, that your treatment is not working right away, instead of waiting a week or more to find out that the bacteria is resistant to the treatment that you're giving. By doing imaging, we can find a lot of things that had not been known before. The knowledge of the disease is really important, and just finding out little things that in the future might have a significant impact in people that are suffering from infectious diseases makes it worth it to spend a lot of hours in the lab.
The WMIC is a yearly meeting for the WMIS, which is the society, and I think that being able to highlight infections would be very critical for multiple reasons. I think one, the WMIS is becoming very interdisciplinary, where we have people who are very good at imaging and developing imaging probes. I think it would be a good forum for people who are in infectious diseases, who have ideas about infections and what they do and understand how bugs operate, to be talking to those people. So the WMIC would be a great forum and an opportunity to do that. I think the WMIC could also be a great opportunity for being able to highlight the current knowledge gaps that we have. So what do we have and what we don't have, and what do we need to get there?
So being part of the IOI group allows us to find out what someone else is doing, collaborate among ourselves. If we have projects that are similar, shared how we're doing it, how we can solve a lot of the problems. In some of the past meetings, I've found out really useful answers from colleagues in different institutions where they had the same issues that I had, and just by talking how we solved it, we found that we could make our work way better. And we just try to find a way to move this field forward. There are great groups doing this research in multiple great universities all around the world. Some people are doing the exact same thing that we're doing. Someone is doing something complementary. So we're not trying to compete. We're trying to collaborate to move this forward and to solve whatever bumps we get along the road together.